E-content on the topic exogenous antigen processing and presentation. The class II MHC pathway for presentation of proteins degraded in acidic vesicles involves the proteolytic degradation of internalized proteins in late endosomes and lysosomes and the binding of peptides to class II molecules in this acidic vesicular compartment. This sequence of events include ingestion of protein antigens into vesicles, proteolytic digestion of antigens in acidic vesicles, biosynthesis and transport of class II MHC molecules to endosomes, association of processed peptides with class II MHC molecules in vesicles, the HLA-DM molecule editing, expression of peptide class II MHC complexes on the cell surface. Ingestion of protein antigens into vesicles. Most class II MHC-associated peptides are derived from protein antigens. Proteins that are ingested into vesicles are most commonly extracellular proteins captured by endocytosis, pinocytosis, or phagocytosis. Different APCs can bind native protein antigens in several ways and with varying efficiencies and specificities. DCs and recognized structures shared by many microbes. Macrophages also bind antigens that are opsonized by antibodies or complement proteins and enhance antigen internalization. Surface Ig on B cells can effectively mediate the internalization of proteins present at very low concentrations in the extracellular fluid. Proteolytic digestion of antigens in acidic vesicles. Internalized proteins are degraded enzymatically in late endosomes and lysosomes to generate peptides that are able to bind to the peptide binding clefts of class II MHC molecules. The most abundant proteases of late endosomes are cathepsins, which are thiol and aspartyl proteases with broad substrate specificities. Biosynthesis and transport of class II MHC molecules to endosomes. Class II MHC molecules are synthesized in the ER and transported to endosomes with an associated protein, the invariant chain, which occupies the peptide binding clefts of the newly synthesized class II MHC molecules. The alpha and beta chains of class II MHC molecules are coordinately synthesized and associate with each other in the ER. The folding and assembly of class II MHC molecules are aided by ER-resident chaperones, such as calnexin. The invariant chain associates with class II MHC dimers in the ER and directs newly formed class II MHC molecules from the trans-Golgi to late endosomes and lysosomes. It also prevents the class II molecules from traveling to the cell surface. Although class II MHC molecules are relatively resistant to lysosomal proteases, the invariant chain is degraded in this compartment, leaving only a 24 amino acid remnant called class II associated invariant chain peptide, CLIP, which sits in the peptide binding cleft. The HLA-DM molecule edits the repertoire of peptides being presented, favoring the display of peptides that bind with high affinity to class II MHC molecules. The displacement of CLIP and its replacement by a higher affinity antigenic peptide in lysosomes are accomplished by the action of a molecule called HLA-DM. Peptides that bind to the MHC molecules with high affinity cannot be displaced by DM. Another dimeric class II MHC-like molecule, called HLA-DO, binds to HLA-DM in lysosomes and negatively regulates the function of DM. DM levels rise but DO levels do not, resulting in more efficient peptide exchange and antigen presentation. HLA-DO functions like a chaperone for HLA-DM. Because the ends of the class II MHC peptide binding cleft are open, large peptides may bind and are then trimmed by proteolytic enzymes to the appropriate size for T cell recognition. Expression of peptide class II MHC complexes on the cell surface. Class II MHC molecules are stabilized by the bound peptides and the stable peptide class II complexes are delivered to the surface of the APC, where they are displayed for recognition by CD4 T cells. Once expressed on the APC surface, the peptide class II complexes are recognized by peptide antigen-specific CD4 T cells, with the CD4 coreceptor playing an essential role by binding to non-polymorphic regions of the class II MHC molecule. Thank you.